It's wonderful to be here. I was in Israel once before, and I think the Hebrew's getting me. Okay. And it's my first time in Haifa. The topic that I talked to Carl about, harnessing genetics in the service of kidney health, is one with which I have no conflicts of interest, and there are many aspects to this. And as a result of the breadth of this particular topic, this introductory talk will cover a lot of different subjects. And I have no conflicts of interest relevant to this. So why should we harness genetics for kidney health? And some of this was touched on this morning. Chronic kidney disease, as currently recently published, is present in almost 13.5% of the world's population, with the range written there, and reduced kidney function in more than 10% of people worldwide. So it's a big problem, and both CKD and GFRs under 60 are, as we all know in this room, are associated with a host of adverse outcomes. Acute kidney injury is also a major problem, and 21.6% of hospitalizations among adults and an incredible 33.7% of hospitalizations among children are associated with renal injury, with high morbidity and mortality. So obviously it's an important topic. So I'm going to talk about where we were with genetics, where we have come, and where I think and others think we ought to be. So by the early to mid-20th century, it was clear that there were rare inherited diseases such as cystinosis and Lowe's syndrome and a number of others that were associated with kidney disease, and the inherited patterns were known, but the genes were not. And there was also a recognition of various cystic kidney diseases and familial nephropathy. And at the time I was a fellow, which was now before, no, it was a little bit after the Six-Day War, but not that much after. It was, we see this, and as a future pediatric nephrologist, I had the various books of children who had genetic syndromes or looked different from everybody else, but why wasn't clear. So I was actually told these various syndromes just happen. It won't happen again. This mother and father don't have to worry. But that turned out quickly to be obvious that it did happen again. And during my training, I saw people with familial FSGS. I saw children with various forms of Acute, which Dr. Groupman will, Dr. Vivant will talk about later, that these just happened, and we know they didn't. So one of the first patients I saw, and I'm now a grandmother as well as a medical grandmother, is this little boy. He had obvious cystinosis, but I was probably the 10th young specialist who'd seen him, and some older ones as well, and nobody knew what was wrong with him. He'd had a bad start to life with meconium aspiration, and he'd been on ECMO, and it was just thought, well, he's not thriving yet. But he had glycosuria, he had obvious renal rickets, and he had a number of problems, but he did have a full renal Fanconi syndrome. And what was known at that point is one replaced what was missing, and cysteamine, which allows cysteine, which is abnormally stored in lysosomes, to get out of cells, it turns out, through another transporter, not through cystinosin, which was discovered as the transporter that's mutant in this condition. So I, like most pediatric nephrologists, was interested in genetic disease early, though, as you heard, I'm more of a hypertension specialist. So By the time I finished my fellowship, there were a lot of breakthroughs in genetics. In 1985, the first renal disease gene, as I think most of you know, PKD1, responsible for ADPKD, was discovered. But it was tedious and slow progress until the human genome was sequenced and high-throughput next-gen sequencing developed. Now, more than 1,200 genes associated with kidney disease and renal development are known. And there's much more we need to know. So where do we need to be? The International Society of Nephrology had a think tank this past year, and they divided into a number of subjects, and one of those was where should genetics go for kidney disease. One was increase awareness that genetics and knowing genetic causes of kidney disease is going to be increasingly important not only for our understanding but ultimately treating kidney disease. That since most of the populations that have been genotyped so far are of European ancestry, that the breadth of what's known about genetics and genomics in terms of data should be really reflecting populations around the world, that accessibility is important, and certainly open data, which I think working at the New England Journal, and you heard a bit about that this morning from Dr. Drazen, is that open data and sharing data so it can be looked at and turned into what we know is important. But that's been standard in genetic research. But usability could improve. So access to the data need to be easier. We also need to improve tools to do functional and practical studies and improve interpretation. This is obvious, and what I'm going to do in the rest of this talk is give a few examples. The idea of personalized medicine is out there on the internet. This is not from an academic source. This was one of the approximately 20 concerns I looked at that would tell you your genotype and tell you your ethnic background, tell you about your mitochondrial genes, and this particular site, the URLs I've listed, are you can find out your risk assessment. You can prevent things and look at what your genes tell you about what you should do and what you should avoid. It will be important because we can help you get your disease detected early before it manifests. And we can give you an accurate diagnosis of your risk factors and provide you a treatment strategy. And you're going to do well if you sign up with us, and we'll help you manage it. And all you have to do is call us, text us, go to our website and input your data, and it won't even cost you that much. And one of the really promising things, as you all know, is that the cost of doing genetic studies is decreasing. It's important for us all to know, and I'm sure all of you in this room know, that there are different types of data that high-throughput genomics, epigenomics, transcriptomics, proteomics, and metabolomics can provide. And I don't think I'm doing well with arrows, but you can probably see this. But certainly the number of genes and structure and function are available through genomics, and there are now increasing numbers of targeted gene panels and sequencing available. The epigenome also is providing more and more tools to look at things, and so on down this list. And there are a number of common nephrological databases, which probably many of you have used, and these are listed here, and provide the opportunity to see what is available, to compare it, what you've found, to look at a paper, and I'll show you an example of that, and see how much more has been found. Epigenetics is important with methylation and demethylation being possible to detect easily. And here I've just put a couple of diagrams. And if you look at the pie charts, what you can see is that in chronic kidney disease, there's certainly evidence for increased or decreased methylation, and that may have important implications for studies that we do. So I'm going to now give a couple of examples of conditions that are genetic and studies of interest, and some of them will touch on work that a number of you have done and talks that will follow. And also, by identifying such targets in terms of genetic abnormalities, one has the possibility of doing drug development and ultimately perhaps rescuing the phenotype. So these days it's possible, and a number of you here have done wonderful studies in this area determining the causes of rare conditions that affect the kidneys. Now, this is one of the earlier studies that, of its kind, that the journal where I work published, and it's the so-called East syndrome: epilepsy, ataxia, sensory neural deafness, and tubulopathy, and turns out to be due to mutations in KCNJ10, which encodes for the KIR4.1 gene, which is a potassium transporter. This was a study on just a few people, and it is clear that these are consanguineous families and that this is an autosomal recessive, and you can see a slightly abnormal brain, and you can also see that there's hearing level loss in the patient who's shown here. What you can see in linkage studies is that on chromosome, shown chromosome 1, there are identical alleles that are in a linked region in all affected patients, and there was a very high LOD score. And this was for family 1, and the single peak is almost 5. And what you see here is this transporter, and it turns out that the R65P is important, and that this is causal for this unusual condition, but it's not so unusual that pediatric nephrologists don't see it. So curve 4.1 does seem to play a major role in renal salt handling with its function, and it may also have some implications for blood pressure maintenance and regulation. Since this paper was published in 2009, there are a number of databases that have included reports of it, and one can go and find that the allele frequency is very unusual for these abnormalities, and the finding has held up. I am now carrying coals to Newcastle, in that a number of you here have been deeply involved with studies of APOL1 variants and kidney disease in African Americans and in various conditions. And I'm only going to touch on it briefly because I think that Dr. Otto will be mentioning studies in Africa on it, and I'm sure you're all familiar with this. Mapping by admixture linkage disequilibrium studies showed genetic differences in African Americans and those African Americans with kidney disease. And as you know, early studies pointed to this area, 22q13, on chromosome 22. And the initial report, as you also know, suggested that MYH9 variants were causing this risk allele to be present. But when a larger area was resequenced and in silico studies were done mining data from the 1,000 Genomes Project, it turned out, as we all know, that the APOL1 gene was involved. And MYH9 was in strong linkage disequilibrium with even more highly associated G1 and G2 variants. The G1 haplotype with 2 missense variants in nearly absolute linkage disequilibrium are present, and the G2 allele has an in-frame deletion of 2 amino acids, shown here, and the association with MYH9 haplotype disappeared after controlling for the APOL1 risk variants. And this is a slide that some of you will know, since it's actually pinched from Dr. Cruzel de Villa et al., which include a number of you, including Dr. Skorecki, show that if you have 0 risk alleles, you can't lyse trypanosomes, but you don't have risk for renal disease. There's a heterozygous advantage with 1 risk allele, but if there are 2 risk alleles, you have a homozygous disadvantage and association with kidney disease. One of the many papers on APOL1 risk variants was one that was published in our journal some time ago, of risk variance, race, and progression of CKD. And this one slide from it indicates that, as you can see here, in patients with diabetes, if you look at the difference in estimated GFR, or you look at patients free from a renal outcome, there are differences that you see between Caucasians and Blacks, and same in patients without diabetes. So using a number of tools, including GWAS, what we get is pathways to discoveries, which I'd like to talk about a little bit. If one does genome-wide association studies with SNP-associated candidate genes, those findings then need to be replicated, and potentially causal variants can be identified, and then generally we have to validate them in a model and consider possible new inventions. But the idea overall is to lead to a change in practice, which takes an ever diminishing time as the tools improve. One of the things that one wants to think about in contemplating studies is how will the payoff be. Obviously, in this corner, if you have a big effect from a variant and a common variant, you're most likely to find something. And I've listed, in addition to APOL1, and there are more diseases for which it's a risk marker, there are markers for membranous. And I've listed some monogenic disorders here and common SNPs, but this is when the effect is lower. So frequency and effect size are key when you consider clinical trials and what one can do with these data to improve kidney health and treatment. I come from an era when we really didn't have genetic information, so if you were doing a study, what you were going to find was if there was some sort of genetic difference that would lead to someone responding, we wouldn't know ahead of time. But there may be in such a group of people a group who have a gene that causes adverse events, and those people won't do well in the trial, or somebody might have a variant with potential efficacy. An example or two of genetics and effective therapy are Hep C and genotype-based therapy and a twofold change in response to PEG interferon and ribavirin, misspelled, and rare mutations such as the BRAF V600E mutation will tell us who with metastatic melanoma will respond, and those with variant respond well. And I think increasingly we will want to do these sorts of things and determine relevant gene variants in patients who come down with various kidney diseases. Genetics may also predict ineffective therapy. This study showed that a variant in C5 affected 3.2% of paroxysmal nocturnal hemoglobinuria patients, and they have a very poor response to eculizumab, so it's better to know in advance. So back to cystinosis. The little boy you see here is now a college graduate working full-time, was on cysteamine since he was about 10 months old, which was when I met him. And unfortunately, despite cysteamine, did need renal transplantation, but he is, relatively speaking, doing well, leading a very full life. And what he wasn't able to have was what may be coming up for cystinosis and some other rare diseases. It was found in the 90s by Corinne Antoniak and her lab that the transporter cystinosin has a number of mutations in its gene, and these lead to a protein that really isn't able to transport cysteine out of cells. And as I said, what happens is with cysteamine, it complexes with 1/2 cysteine, and that allows it to be transported out of cells. However, in the 15 years ago or so, since cystinosin has been discovered, a host has been learned about this disease. We know the pathways of cystinosin and know that inflammation is associated with that transporter, and know also that the mTOR pathway is involved. And additionally, a number of cell lines have been developed that have allowed multiplex drug screening, with some candidates that seem to help patients store less cysteine in cells and also improve the Fanconi syndrome. And these are compounds that really were already FDA approved and just found in vitro. In this particular study, the patients are mice. Stephanie Shirky and colleagues have created a model of cystinosis in the mouse, and what you can see here is that the animals don't do very well, and the lines that aren't going anywhere or going up are storing cysteine in these different mouse tissues. However, using hematopoietic stem cells, these mice can be rescued and actually not develop renal failure, not develop crystals in their eyes, not develop the intestinal effects of this condition. And there's presently a protocol ready to launch to actually do this in affected patients. And so we'll see how that goes. But I think in my lifetime, I will have seen something change from, we see this, this child isn't going to live past 10, to the fact that most patients with cystinosis do live into their 30s and 40s. I've met a few in their 50s, and now there is the hope that the disease can be cured. So this just shows the reverse direction in the kidney in these mice. It's also possible with open data, as I hinted before, to look at a study and go to the database, in the 1,000 Genomes database, and find more loci that support kidney function. In this particular study, there was a meta-analysis done, and this found that there were more than 50 loci that were common variants that appeared to associate with kidney function, and using more complete reference sets and imputations, loci were found that hadn't been found before. And ultimately, in this particular study, there were 10 new genes that really do seem to be important for kidney function. And these are just shown here in this Manhattan plot. As I said earlier, drug testing is going to be possible by delineating molecular targets. In this particular review, Santos et al. Pointed out the many different classes that seem to have effects on different organs, and as a result of the mapping in this study, and I'm not going to go through it because there were too many figures to show in my short time, there are a number of targets that have been found. Another example about looking at patients who already are known to have an established genetic variation may be important in looking at a trial. This particular trial was published last year in the New England Journal, and as nephrologists, we all see people with Fabry's disease, and we mostly treat them with Fabrazyme. And people develop antibodies to it, or they can't get it, and this is a different approach. There's a pharmacologic chaperone, megalostat, that unloads cells, and when this study started, and this is like most studies, a flawed study, when they began, the assay that was used didn't do anything to differentiate which patient would respond. But they developed an ELISA that showed abnormalities and a mutant protein that would respond to this chaperone, and indeed that seemed to increase the power. And although the study is small, the total number of patients needed, if you can cull out who will respond, is very helpful. And in this, you can see a change from baseline, and the blue is those on megalostat from the beginning, and this line then goes down when the ones who had been on placebo got megalostat. In our future, as nephrologists, certainly CRISPR-Cas9 engineering was going to make a lot of studies possible. It's certainly possible, given this technology, to target any gene and then to design what you want to put into cells. And already in vitro with renal cells, people are doing this, but I don't yet, and maybe somebody else does and can tell me, provide a CRISPR-Cas9 example in a human being for kidney disease. I've found several for different conditions in animal models. This is a review article that talks about the promise of CRISPR-Cas9 for kidney research, and so ultimately therapy. One can design an sgRNA so that strains can be studied without having to get just the same background. And this is species-independent genome editing. In this particular trial, species and strain are mixed up, but that may be helpful in establishing new suitable models for kidney research. Excitingly, CRISPR-Cas9 may be helpful in doing xenotransplants, with the idea that one could potentially make some engineering changes in pig kidneys and then transplant them to people, and also do genetic therapy and in vivo gene delivery very specifically. Additionally, inducible pluripotent stem cells have a promise, and this is a particular just schema of what might work. And as you can see, you could do a punch biopsy, transduce the cells, decide what to culture them in, and then within a few weeks pick colonies and give them back. Whether this will be used soon in renal disease, I don't know, but I think a number of people, including some in this room, may work on that. What we definitely can do right now is bring urine and also kidney biopsies to the molecular age, which isn't done as routinely as it might be. Certainly, when a kidney biopsy is done, there's sectioning and routine diagnostic studies and looking at the clinical record, looking at the histology, and perhaps post facto looking at the molecular phenotype and doing clinical correlation. One can certainly microdissect a renal biopsy or do laser capture and do many studies, and this is actively being done. Additionally, body fluid-based analysis is also possible, and presently there are some cohorts being looked at for intrarenal candidate markers for acute renal injury and for progression. And what one can do is take cells from the urine and extract DNA or RNA, and that's being done commonly. Ultimately, validated markers can then identify relevant signaling pathways, as you know, in silico, and then provide treatment targets, and intrarenal messenger RNA and urine protein correlations and other correlations can be done. And ultimately, the idea is to have non-invasive biomarkers and to have studies that show outcome and compare outcome with classical renal biopsy methods and markers as compared to molecular markers, and really look at management with a large enough patient sample and replication cohorts for it to be believable. So genetic testing, I think, is really important. I would say 5 or 6 years ago, my own opinion was maybe we shouldn't always do this. I now think more often than not, genetic testing may provide definitive causes for a given condition, and it would facilitate accurate and specific genetic counseling, avoid unnecessary diagnostic tests, procedures, and treatments, may allow early treatment before any symptoms are present, may detect apparently unaffected family members who actually have the condition, and may facilitate monitoring if a definitive cause is found. And I think that management on a per gene basis is something that is coming in nephrology, and I think we should support doing it as much as possible. So with this quick overview, I thank you very much and look forward to the talks of others.
Questions? Any questions in the audience? We have time for one question. So I would like to ask you, Julie, what do you see as the most important bottleneck for genetic testing in the white population for any child who comes in? Because I know what the bottleneck in Israel is. It's the policymakers, decision makers, it's funding. There is a basket, a very limited one, and many sick children are not eligible to undergo genetic testing just because of the policy. What is the state of it in the United States, in the Western world? Where do you see it goes from here?
Well, I probably should have said that it's not so easy in the U.S. either. We, with our multi-payer system, have to argue at great length with insurers to allow testing to be done when it's a good reason. I think that with the cost of genetic testing and the cost of studies going down, that this will be, at least assuming no catastrophes in the world, something that will be much more feasible to do in the next 5 years. But yes, it's a big problem.